investigation is when you investigate what it is. What is a microscope, right? So we have two learning targets. Say learning targets.
Learning targets.
So your first one, what you're going to be doing, you're going to be identifying parts of a microscope. Say identify.
Identify.
What does identify mean? Christian?
Figure out.
To figure out. Good. So say I can. Identify. Parts of a microscope.
Parts of a microscope.
Good. Your second target, after we identify the parts, you're going to analyze a plant specimen. Say analyze.
Analyze.
Who can raise a quiet hand and tell me what analyze means? Zio.
Like to observe.
To observe, to look closely, right? So you're going to look closely at a plant specimen because we're learning about photosynthesis. So that's what we'll do after we identify parts of the microscope. So these are called light microscopes. Say light microscopes.
Light microscope.
They have a light on the bottom. So let's look at the different parts here, just so we can read the vocabulary words. Who can raise a hand and read this one? Hardeep?
Course. Focusing. Mechanism.
Say mechanism.
Mechanism.
Good. High five in the sky. This one? Everyone?
Arm.
The arm is where you hold it when you carefully carry it to put it back or to take it out. Fine focusing mechanism. Say fine focusing mechanism.
Fine focusing mechanism.
Good. Then you have, so the fine focusing mechanism on your microscope, it's the smaller one. That's why it's called the fine. So can you touch the fine focusing? And my friends at home, if you're on your virtual microscope, you can start clicking it to look at the different parts. Okay, your virtual microscope. Good. So then we have the base. Say base.
Base.
The base is the bottom. So I was showing you before, you're going to hold it by the arm in one hand, and you cushion it under the bottom by the base when you carry it. This is called the body tube. Say body tube.
Body tube.
Point to your body tube. Good. Up top, you guys have a lens. Point to your lens. That's where you look through the lens. It's like contact lens, right? Revolving nose piece. Say revolving nose piece.
Revolving nose piece.
So take your revolving nose piece and gently turn it. Gently turn it. Homies, my friends at home, you can manipulate your virtual microscope, I think by clicking it. It might be able to turn. But you can just check out the different parts. Okay? Malik has his own microscope at home, so he's working with identifying his parts. Objectives. Say objectives.
Objectives.
So objectives are the three lenses. Okay, look at your three lenses. What numbers do you see on each lens? Who could tell me? David?
0.10, 0.65, and 0.25.
So notice how math is connected to science, right?
Yes.
The lenses are magnified and they use decimals. So one of the lenses is 40 times. Can you find the 40 times? It's the blue one. What do you think? Okay, and then turn it again. Who could tell me what the yellow color is? Cheyenne?
0.25.
0.25, okay. Does it say any other number on there? Yeah.
0.17.
Okay. And what do you notice about the red magnifying lens? The red one. Layla?
It says 0.10, 0.17.
Okay, good. So there's 40 times, and then you should have, so you have smaller lenses. The other two are smaller. So the 40 times we learned in math, right? You times it by a power of 10, it's going to be bigger. So it's going to get magnified. Say magnified.
Magnified.
So when you're looking at a tiny, tiny, tiny cell that we can't see, the lenses help you see it because it gets magnified. Make sense?
Yes, it does.
Okay. Mechanical stage, say mechanical stage.
Mechanical stage.
Find the stage on yours, it's the black platform. That's where the slide goes. Then you have the clips, touch the clips, the silver clips. So you have the stage is the black platform, and then you have the clips. So Cheyenne is gently moving her clips. So that's how it holds it into place. Remember the button in the back? I said you could tap that when you put it under. Gently, gently, good. And then we have a substage, which is the pieces underneath. We could just call it substage, say substage.
Substage.
And then at the bottom, there's a mirror. I don't know if you can see it on yours, but the mirror works with the light to reflect the image. And that's how you're able to see it. Okay, question so far? A question? Yes.
I noticed that under the base there's like a wheel pattern.
Yes, so there is a wheel, and it's also part of like a way to focus it. Every microscope's different, right? So these are all the same, but Malik's at home might be a little bit different. Yes, Malik?
Mirrors don't get sun, right, when you're outside?
Say that. So do you have a light microscope? Does yours have a light?
Yeah.
Yeah, so the mirror reflects light to help you see the image clearly. Yep, yeah, okay. So that's what we were just saying, what's a light microscope? A light microscope uses visible light and series of lenses to magnify an object or make it bigger. And we kind of went over the different parts. These are the different types of microscopes that you might see. Maybe if you go to middle school and high school, they might look different. So we were talking about the different lenses, right? So again, there's a red objective, then the yellow is the medium, and then the blue is high power. Okay, the highest power for our microscope is the blue one. Make sense? And you have to be careful when working with the blue one, because the blue one could get very close to your slide when you're moving the stage. Okay? "100 times is medium power, 400 times is the blue power." It's kind of cool, right? The red one is, I think it's low power. It's 40 times. Okay, we went over how to carry a microscope, so we're going to practice that when we put them back. So how we use it. Who can raise a quiet hand and read this? How would you start with looking at your slide? There's a way to do it. There are steps to do it. Ariana?
Start on red objective. Make sure the objective above the mechanical stage is set to low power.
Right, so everybody turn your knob to the red one, because that's where you're going to start when I do give you a slide. They want you to look through that piece first, you'll adjust your knobs, and then you'll work through the different magnifications. Then what do we do next? Loud and proud. Haley.
Turn on your microscope and put slide under the low power
Yep.
objective
Yep.
so that the light passes through the segment.
The specimen. Say specimen.
Specimen.
Good.
You can see the standard color.
Right. So you're going to get your slide. The light's going to pass through it, and you're going to be able to see. It's called a specimen. We're going to look at a plant cell. What's step 3? Step 3, Ryan?
Recommend using a large knob. This moves slowly.
first you start on the red one. Then they want you to use which knob? What was the vocabulary word we called it? Instead of large, what's the vocabulary word that we called it? It's on your chart at your seat. You can use your chart. Zio?
The fine focus?
The fine focus is the smaller one. There's another one. The fine focus is the smaller one. It finally makes it clear. Cassidy? Yes, say coarse focus. Good. So first, find your coarse focus. It's the larger knob. Good. Then we use the fine focus, like Ziomara said, and it's the smaller knob. And it's just to really focus in on the specimen more clearly. My friends at home, if you have the virtual one, you will notice the two knobs, and then it should zoom in for you, and you'll see the difference when we get into that, okay? After you work with the knobs, then you can start switching to the other ones. Is the yellow one would come next. So you would do red, big knob, small knob, yellow. You can adjust the knobs a little bit, and then you can try the red. So I'm going to skip through these a little bit so we can start. Oops, going ahead. Okay, so here's what I want everyone to do first. Before we look at a specimen, I want you to work on your first learning target. So your first learning target, who remembers what it was? What are we going to label on our worksheet in our microscope packet? Brianna? Yeah, high five this guy. Good job. You're going to label the parts of your microscope now. So my friends at home, you have your microscope packet. Great. So you guys can start just labeling it. It's the picture, the first picture. The first picture with the arrows. Okay? You can use your chart to help you with your vocabulary. Good job. So in about 7 minutes, we'll hopefully go on to the second part. So flipping to the front, flipping to the front. So you're working on your worksheet. Great job, Michael. And when you're done labeling, you can put your hands on your head.
Miss Morris.
Yep, 1 second. Okay, so now for the second part of today's lesson is you're going to analyze a specimen. Say analyze a specimen.
Analyze a specimen.
And the specimen that we gave you were a stem, say a stem,
A stem.
and a plant leaf.
And a plant leaf.
Why are we analyzing stems and plant leaves? Ariel at home, can you tell us why we're looking at those? Why are we looking at plant cells? What are we reading about and what are we having a test on next week? Ecosystems, and we learned that, Malik, you want to add on to what she said?
And we're learning about photosynthesis.
And good. Good, high five in the sky. We learned about photosynthesis, and so now we're going to look at the cells, because we did study the cells in our textbook, okay? So remember the steps when you're gonna look at your specimen, everybody starts on red, and we're carefully moving the lenses, right? And you're carefully moving the knobs. All right, so you can explore your plant specimens. I'll give you about 5 to 7 minutes. Think about what you notice, make observations. You could turn and talk to your partner about what you see from a distance. All right, go ahead. You could go, start. Make sure your light is on. Good, Cheyenne. Keep working with your knobs. You may not see it right away. You may not see it on the on the red.
I see it.
You may have to magnify it.
Oh, I see some.
Use the coarse knob and then the fine knob. Good job, Brian.
Oh, it's really cool.
Green brick with fingerprints on them.
Say that again, Julia.
The plant cell kind of looks like green brick with fingerprints on it.
Yes, agreed. Thank you for sharing.
Can I sure don't mind with the onion leaves?
Yeah.
Okay.
It looks like little bamboo sticks.
Layla says hers looks like little bamboo sticks. So when you magnify a piece of a leaf or stem, that's what it looks like inside. So Layla is sharing her screen with what she sees. So Layla, not Layla, Ariel. See, Ariel's sharing what she sees on her virtual one. Pretty cool, right? I put the link so you guys could do that if you want at home. So Ariel, tell me what you are observing.
Not specifically like an onion leaf. I'm gonna zoom in a little bit.
This is awesome.
Looks like dark purple dots.
Yeah, dark purple dots. It looks like the image in your science textbook. Do you remember that image when it was like zoomed in on the plant? And the
Yeah.
parts were labeled? So
Yeah.
some of our vocabulary words are what you're seeing. Oh yeah, look, hold on everyone, look. That looks like your textbook.
Yeah.
Remember? From your readings? Awesome. You guys are doing a good job.
Oh, I see some.
You do? So then if you think you see something, then work with the fine knob. Good job. Take your time. Great job, Ariel.
I can see the vein.
You can see the veins? Do you have a leaf?
I can see little cracks in them.
It's really neat.
What do you notice? Or what do you
Cracks
observe?
in between?
Cracks in between? Cool. Layla, what do you see?
I see, like, it looks like little bamboo sticks, and I can also see the vine.
Cool.
It's really nice.
Did you get it yet, Sia? Okay, keep trying, right? Use your knobs.
Oh, and I'm like the corner of mine. It's so pretty, right?
Great job.